Hey guys, welcome back to my channel. So today I'm going to give you the Biostatistics 8th semester 10 marks question and answers as per RGUHS question bank. So today I have took the third unit for Biostatistics 10 marks question and answers. So these question I have taken from the question bank, that is biostatistic question banks. If you need the question bank, so please do comment down below. Definitely I'm going to give you the question bank of biostatistics. So I'll start from the third question. So in the question bank, this is the third question. Explain the different phases of clinical trials, and these are the questions for third unit 10 marks as per RGHS question bank. So I have listed out for the third unit, so you can easily note it down. So I'm going to start with the first question, or it may be third question. So explain the different phases of clinical studies or clinical trials. So the clinical studies, after submission of IND application, on reviewing the data submitted therein, if there is no any objection by FDA, so within 30 days the work described in the IND may be begun. So these processing clinical trials to qualify the drug for sale to market, which is classified into three stages, that is phase 1, phase 2 and phase 3. So the post approval trials fall into the phase 4. So the phase 1 clinical trial, that is human pharmacology. In phase 1 clinical trials, the candidate drug is tested in the peoples for the first time. So these studies are usually conducted about 20 to 100 healthy volunteers, as typically they take 6 to 9 months. So the phase 1 studies are designed to survive safety and tolerability of the candidate drug in the humans, where researchers look the pharmacokinetic of the drug, how it is absorbed, metabolized or eliminated from the body. And they also study the drug pharmacodynamic to check the desired effects for the side effects, and these closely monitor the trails of designs to help research to determine the safe dosing range. If in these series no serious safety effects are found, the investigator may continue to the phase 2 trials. So in phase 1, complete plan of experimental work for phase 2 has been submitted to FDA. Within 30 days, the FDA makes no objection, investigator may be begin phase 2. So the phase 2 clinical trials, it is also known as therapeutic exploratory trials. So the phase 2 studies are designed to determine the effectiveness and the further design to safety of the candidate drug in humans. So depending upon the type of the investigational drug and the condition it treats, this phase of development generally takes from 6 to 3 years. So the testing is conducted with 100 to 500 patients which is suffering from the conditional that investigational drug is designed to treat, where these testing determines the safety and the effectiveness of the drug in treating the condition and establish the minimum and maximum effective dose. Most of the phase 2 clinical trials are randomized or randomly divided into the groups, one of which receives the investigational drug, one of which gets the placebo that containing no medication, and sometimes a third group that receives a current standard treatment which the investigational drug can be compared. Whereas phase 3 clinical trial, or it is also known as therapeutic confirmatory trial, third phase, that is clinical trial, it is also known as therapeutic confirmatory trials. You cannot see, so that's why I am explaining you. So phase 3 is clinical trial, therapeutic confirmatory trials. So in phase 3 trials, researchers studies the drug candidate in the larger number, about 1000 to 5000 of the patients, to generate the statistically significant data about the safety, efficacy and overall benefit risk relationship of the drug, where this phase of research is a key in determining whether drug is safe or effective, or it is also provides the basis of labeling instructions to help or to ensure the proper use of the drug. Example, information of potential interactions with other medicines. In phase 3 trials, there are both cost list and long list trials. So depending upon the type of the drug candidate and the condition it treats, this phase usually require 1 to 4 years of the testing. During the phase 3 trial and even in the phase 1 and phase 2, researchers are also conducting many other critical studies, including the plans for the full scale production and as well as the preparation for the complexes application required for FDA approval, where the second question, that is, discuss the protocol for an experimental study design. So the experimental study design is an experimental research design. So these are concerned with the examination of the effect and independent variable on the dependent variable, where the independent variable is manipulated through the treatment or interactions dependent variable, where the effect of these interventions is observed on the dependent variable, or the experimental research design is a powerful design for testing a hypothesis of causal relationship among the variables. So this is the box, the method of the data analysis, where the element of research design consists of method of data analysis, tools and materials of the data collection, the approach and the time and play of data collection, quantitative, qualitative or both, and population, sample and sampling treatment, where plus, with or without consumption framework. So these are the two broad classification of experimental design, such as formal experimental design and informal experimental designs. So the formal experimental design, study design, so the designs after the relatively more control and the use of precise statistical procedure for the analysis of formal experimental, which are classified as completely randomized, number second, randomized block, number third, Latin square, and number fourth, factor design. So the informal experimental study design, before and the after without control design, where the level of the phenomenon after the treatment and the level of the phenomenon before the treatment, where the test area is equals to level of the phenomenon after treatment, that is Y, which included treatment induced level of phenomena before treatment, that is X. So the treatment effect for before and after without the control design is equals to Y minus X, and after only with control design, so the value of the dependent variable in the test area, so the value of the dependent variable to be controlled area. And in these design, two groups or the areas are selected and the treatment is introduced into the test area only, where the dependent variable is then measured in the both areas at the same time. Treatment impact is assessed by the subtracting and the value of the dependent variable in the control area from its value in the test area, where the two group or the two area are selected and the treatment is introduced into the test area only. So before and after with control. So in the previous we have studied before and after without control design, and in this before and after with control design. So the change in the dependent variable in the test area minus changing in the dependent variable in the control area, where the test area time period 1 level of phenomena before treatment, that is x minus y, and control area, that is level of phenomena without treatment, that is A, and level of phenomena without treatment, that is Z. So the treatment effect for before and after with control design is equals to y minus x minus z minus a. And coming to the next question, so explain types of observational studies, where the observational studies are important type of study design, and it is fall under the category of analytical study design, where the observational studies are the one where the scientists are observe the effect of the risk factor, diagnostic test, treatment or other interventions. So in these studies use samples to draw the conclusion about the population when the researchers do not control the treatment of independent variable. This study researchers only absorb the subject and do not interfere to influence the outcome. Usually the scientists conduct the observational studies when it is impossible or unethical to assign the study participants and to experimental the groups randomly. The main basic type of observational researchers are descriptive research, where the main aim to analytic the research is to test the hypothesis about the association between two or more variables. So there are many studies to design that fall under the umbrella and the descriptive study design, and that included the case report, ecological studies, and case series, cohort studies, cross-sectional study, and as well as case control studies. So the types of the observational studies in that case report and case series, ecological studies, cross-sectional, case control and cohort studies, where number 1, that is case report and case series. So the detailed presentation of a single case or handled of the case. Generally the report of a new or unique finding, where example the previous undescribed discussed, where unexpected link between the discussed and unexpected the new therapeutic effect where the adverse event occurs. So the case series experiences of group of the potent with a similar diagnosis and assess the prevalent diseases, where the case may be identified from a single or a multiple sources. So the generally report of a new or a unique condition which may be only realistic design for rare disorders. And number 2, that is ecological studies, where the ecological studies are based on the analysis of aggregated data at groups level, which do not involve the data into individuals. So typical examples included the study that examined the patents and the drug use over time, where the comparison of the use for non-steroidal anti-inflammatory drugs and COX-2 inhibitors in the Australia and Canada. So they describe the association between the drugs and the outcomes, such as changes in the rates of upper GIT, hemorrhage after the introduction of COX-2 inhibitor, where the demerits for the ecological studies are individual level data are not presented. So number 3, that is cross-sectional study, where the cross-sectional studies collect the data at a single point in the time for each single individual, but the actual data collection may take place over a period of time or more than one occasion. So there is no longitudinal follow-up and individuals which provide a profile of a population about the interest which may be broad. Example, Australian Health Survey undertaken intimating the Australian Bureau of the Statistics. Number 4, that is case control studies. So the case control studies focuses on determining the risk factor for an outcome of interest such as diseases or a drug adverse effect that has already occurred. So the steps involved in the case control studies: number first, two groups of participants are assembled, those who already have the outcome case and those who do not have the outcome case, and who are often matched to the case to make them similar and reduces the biases, where the second, the data on previous exposure to be selected for the risk factor and are collected, where these are composed to see if the risk factor are more criticizes were common among the case versus control. Multiple risk factors can be studied, but each case control study can involve only one outcome, where number 5th, cohort study. In the cohort study, an outcome or diseases or the disease-free study population is the first identified by the exposure or the event of the interest and allow in the time until the disease or the outcome of the interest occur, where the exposure is identified before the outcome. Cohort studies have a temporal framework to assess the causality and potential to provide the strongest scientific evidence. So the past, present and future, where the study starts. So reprospective cohort studies looking back in the time, prospective cohort studies looking forward in the time, where the study population or the selected samples are divided into two groups, that is exposed group population and unexposed group population, where the exposed group population is also divided into two types, that is condition develops and condition does not develops, whereas the unexpected group population is also divided into two and it is have the same, that is condition develop and the condition does not develop, where the retrospective and the prospective cohort studies. And next question, that is explain the chi-square test. So the chi-square test is a non-parametric test which does not based on any assumption or the distribution of any variable. So in general, the test we use to measure the difference between what is observed and what is expected according to an assumed hypothesis is called as chi-square test. So it is also denoted by the sign of x square, where the following formula for the chi-square test is x square is equals to summation of absorbed minus, that is expected whole square divided by expected, where the chi-square test is calculated by dividing the square of the overall deviation in the absorbed, expected and frequencies by the expected frequency, where if there is no difference between the actual and the absorbed frequency, the value of the chi-square is 0. Where there is the difference between observed and the expected frequency, then the values of the chi-square would be more than 0. And the properties for the chi-square distribution, the square of the standard normal variables is a chi-square variable with 1 degree of a freedom. That is, if x is normally distributed with mean or a mu and the mean deviation, that is standard deviation, that is sigma, then the formula is x minus mu divided by standard deviation, that is rho, and is a chi-square variant x square. So with one difference, the distribution of the chi-square depends upon the degree of the freedom. There is a distribution for each number of degree of freedom. So the mean of the x square distribution is equal to the number of the degree of the freedom, that is n, and the variance is equal to 2 times of number of the degree of the freedom, that is the variance of the x square distribution is equals to 2n, and the mode of the x square distribution is equal to n minus 2, and the lowest value of the chi square is 0, and the highest value of the infinity, that is x square, greater than or equals to 0. The test can be used in goodness of fit of distribution, test of independence of attributes, and as well as test of homogeneity. In that, number one, goodness of fit of distribution. So the goodness of fit is the test which is a statistical hypothesis test to see how well the sample data fit a distribution from the population with a normal distribution. So the goodness fit of establishes our disparity between the observed value and as well as those that who expected the model in a normal distribution case, where it enables the US to see values and assume the theoretical and also x square is less than the table value. And this is the example for number second, that is test of independence of attributes. So x square test enables to explain whether or not to attribute are associated, testing the independence which determines the two or more observation across the two population that are dependent on each other, whether one variable helps to estimate the other. So the two attributes are independent or not, that is associated if we calculated the value that is greater than the table value, so which you reject the null hypothesis. And what type of data the chi-square test is performed. So the data used for the chi-square must be quantitative in the form of frequency, which must be absolute or not in the term of relative, and the total number of observation are collected. This is the large and should not be in the random basis, where the observation makes the sample. This test, which must be independent of each other, and the expected frequency of any item or a cell must be less than 5. So the frequency of the adjacent may be pulled together in order to make it more than 5, and the observation must be independent of each other, and the data must be in the form of frequencies. So this is the question that arises in the question bank. So this is the problem which I have given already in the biostatistics first unit, which I gave you previous video. And the next question, explain the concept of DOE. So what was the question? What is QBD? Why DOE is essential and in the QBD development process. So explain the concept for the DOE. DOE is design of experiment. So DOE is a process optimization technique that relieve the planned experimentation and the statistical analysis of result, where the DOE makes the control changes to input variables in order to gain the maximum amount of information on causes and effect of relationship with a minimum sample size. So the design of experiment is also referred as a design of experiments or experimental design. So the importance and the basis of the DOE, which reduces the designs, development, new product and the process, improve the performance and improve the reliability and achieve the product and process robustness. So DOE or the design of experiment is a branch of applied statistics that is used for conducting a scientific studies or a system or a process in which input variables x1, x2 and x3 are maximum, where the manipulated to investigate its effect on measured response variable, that is y, y1, y2, y3, and it is one of the important data collection and analysis tool that can be used for the planning, conducting and analyzing the interpreting controlled test in the different experimental situation. So the DOE can be a tool which is used to situations for designs of comparisons, variables, screening, transfer function, identification, optimization and robust design, where the DOE can be performed in the analyzed with the help of a suitable statistical software. So DOE software can be used for the setting of analyzing generate factorial, two-level factorial, fractional factorial, flanked Berman design. It is also used to visualize the response surfaces by rotating 3D plots. Today, DOE is a software which is most valuable tool for the engineers, scientists, biologists and the new drug developers. So the computer softwares are also available for designing or experiments from various software companies, and it includes the package in the important mini tab design, expert statistical SAS, statistical graphs, Microsoft Excel, etc. So the mini tab is a statistical are most popular packages that are equipped with the user friendly interfaces and very good graphic output. And the next question, that is role of QBD in the pharmaceutical development. So the systematic approach to develop that begins with profound objective and emphasizes product and process understanding and process control. So it is based on sound science and quality risk management. So the key aspects for QBD: the target product profile, critical quality attributes, risk assessment, design space, control strategy and life cycle management. Then the role of QBD: designing the development formulations and the manufacturing process to ensure a profound quality, to achieve the meaningful product quality specification that are based on the clinical performance, and to increase the process capability and reduces the product variability and the defect by enhancing the product and process design understanding and control, more efficient use of development time and cost, to increase the product development and manufacturing efficiency, and to enhance the root causes analysis of post-portal changes management, better understanding to the process, less batch failure and allow continuous improving in the products manufacturing process, return of investment and most efficient and effective control of change. So the experimental design which is used in QBD is factorial design, fractional factorial design, central composite design, box Benkin design, flanked Berman design, mixture design, 3:2 factorial, 2:2 factorial and 2:3 factorial design. So how a QBD based on the development, which is better explained in steps involved in it. So the data driven approach which is used for the QBD helps to see in the progressive manufacturing and as well as the environment. It gives the drug develop a better understanding of manufacturing processes. So decrease the amount of batch failure, gives more effective control to change and also provide a higher return on investment. So the steps involved in the QBD: target of the product, critical quality, links MAS and PPS to QCA, establish design space, establish control strategy and assessment, where the drug target to the product quality profile. So it is defined as a prospective and the dynamic summary of the quality which is characterized by the drug products and ideally will be achieved to ensure the desired quality and thus the safety and the efficiency of the drug product is received. And critical quality attributes, where it is a physical, chemical and biological and microbiological property or the characteristics that should be within the appropriate limit range or distribution to ensure the desired product quality. Material attributes, it can be on experiment QCM, raw material starting material QAC, and drug substance by CQA, etc. And a material attribute can be quantified and typically fixed, where the design space, the multidimensional combination and interaction of the input variables and processes parameters that have been demonstrated to provide an assurance of the quality. Design space which is proposed by an applicant and it is submitted by the regulatory assessment and approval. And the control strategy, where lastly drug product material, the risk assessment. And the next question, why are DOE essential in QBD development process? So DOE is one of the element of QBD approach which is discovered by Ronans Fisher in 1925. So the experimental designs play an important role in the new product design and manufacturing processes, where the development and the manufacturing processes is combination of operation machines, methods, staffs and other resources that transform some input to an output. So DOE is a methodology for supplementary applying the statistics to these experimental studies using a graphical or a mathematical approach. So DOE is one of the quick and cost effective method to optimize the product and processes. DOE methodology: choose experimental design, conduct randomized experimentation and analyze the data, create the multi-dimensional surfaces model. And describe briefly the interventional study design. So the answer and the list of the elements that need to be incorporated in a clinical protocol. So the answer for first is definition. Research reports is a condensed form of a brief description where research work can be done for the researchers. It involves the several steps to present the report in the form of thesis or disasters. So in that the different types of report writing are available. In that the number 1, logical analysis and preparation of outline. Number 2, writing the rough draft, listing the references and preparation of final draft. The importance and significance for report writing: monitor and control operation, implement the policies and procedure, documentation, analyze the information and make decisions, and comply with regulatory framework, implantate policies and procedure. Where lastly the question, how will you design a clinical study methodology? Explain briefly. So the clinical study design is a formulation of trials and the experiment as well as the observational studies in a medical and a clinical and other types of researchers which involving human beings. So the goal of the clinical study is to assess the safety, efficacy and the mechanism of action of the investigational medicinal product or procedure or a new drug or device that is used in the development but the potentially not yet approved by a health authority. So it can also be investigated by a drug device or the procedure that can has already have been approved but still the long-term effect or cost effectiveness. So the clinical study design generally classified into 2 distinct types or experimental studies and observational studies. So the basic concept of experimental studies is to affect the intervention, and the experimental studies the risk factor, exposure of interest and control by the investigator, where the research that observational makes no attempts to intervene in the study sample for exclusively investigation purposes. where the observational studies types include wide ranging from the traditional epidemiology, cohort, case control, case series, experiments, etc., where the clinical research involves the analyzing and the association between two or more variables and the variables that are classified as dependent and independent variable. So the outcome for the type of the design, so clinical research study design, analytical, experimental and observational. It is divided into uncontrolled and controlled, where observation is divided into cohort, case series and cross-sectional, where the control trials has divided into two, that is non-randomized and randomized. And the outline types of design: treatment studies for the randomized control trial, blind trial, non-blind trial, adaptive clinical trial, non-randomized trial, that is also called as quantity experiment, and interrupted time series design, where the observational studies, descriptive case report and case series. So the analytical cohort, prospective cohort and retrospective cohort, nested case control studies, community survey, time series studies, case control studies, cross-sectional studies and lastly ecological studies. So here we have completed the third unit Biostatistics 8th semester 10 marks question and answers as per RGUHS question bank. So please do like, share and subscribe to my channel.